one scientist, there was a one scientist, the name was Friedrich. In 1869, he isolated nuclein protein. Okay, that nuclein protein he has isolated from the nuclei of pus cell. Then after some time, they have just observed that particular nuclein having some acidic property, and that's why it became like a nucleic acid. Okay, this nucleic acid, which having a vital role in genetic information, this nucleic acid was discovered by Friedrich in 1869, who isolated nuclein. This nuclein he has isolated from the nuclei of the pus cell, which having some acidic property. That's why it became a nucleic acid. So the next thing, this nucleic acid having a mainly three basic unit. The first one is a base, sugar and phosphate. In this nucleic acid, we will read regarding the DNA and RNA. So DNA and RNA both having a one nitrogenous base. So you have to learn these three unit. That one is nitrogenous base, sugar and phosphate. Nitrogenous base are of two types, purine and pyrimidine. This one sugar, we will discuss here the biosynthesis of purine and pyrimidine base. The next one is sugar. Sugar is of two types. That one is ribose sugar and deoxyribose sugar. So as the name indicates, this one ribose sugar, it is found in our RNA. Okay, a ribose sugar is found in RNA. Deoxy, as the name indicates, deoxy, so it is found in our DNA. And DNA full form is deoxyribonucleic acid. RNA full form is ribonucleic acid. The next thing, if we see based upon their name, we have to see the structure, chemical structure of ribose sugar and D-ribose sugar. So in the further slide we will see, but here we have to observe that in the ribose sugar there will be no hydrogen at one position, but in this deoxyribose sugar, what will be happen? Sorry, in this ribose sugar there will be OH group as second or third position, but in this deoxyribose sugar, as you can see, deoxy means removal of oxygen, where oxygen will be removed and only there will be one hydrogen will be added. And then the next thing is phosphate sugar. Sorry, there will be phosphate will be added in each DNA and RNA unit. Now the next slide. Next slide is function of nucleic acid. As you all know very well about this one, DNA and RNA. DNA having a role in the hereditary characters, or we can say this one DNA or this one nucleic acid having, they can control enzymatic reaction also. They also a mediator of a cellular processes, and they may form a portion of many coenzyme too. That one is NAD, NADP. The next one, this one RNA. This one RNA is used to make a protein. This RNA, it is divided into three parts. That one is messenger RNA, tRNA, and ribosomal RNA. Okay, messenger, transfer, and ribosomal RNA. So messenger RNA, as the name indicates, messenger RNA, it means it will just transfer the message from RNA to DNA. The next one is transfer RNA. It will transfer the activated amino acid to the site of protein synthesis. Okay, transfer means it is used to transfer the amino acid for the synthesis of protein. Next one is ribosomal RNA, which is found in ribosome, and it is responsible for the stability of messenger RNA. Now, this one, as I told you, this one nitrogenous base are of two type, purine base and pyrimidine base. So firstly, we should know the structure of purine and pyrimidine base. Pyrimidine structure is like this. They having a, on the first number and on the third number, they having two nitrogen atom. This one is called as pyrimidine. Pyrimidine ring, where there will be on the first and third number, there will be two nitrogen atom. This one is called as pyrimidine. And the purine, we're having this one pyrimidine, and this one is imidazole. There are two moiety. Purine consists of two rings that you can see here. First ring is pyrimidine moiety. The next ring is imidazole moiety. In the imidazole first and third number, there will be nitrogen atom, and in the pyrimidine ring first and third number, there will be nitrogen atom. We can see in the purine structure, all over there will be 4 nitrogen atom. So we should know very well, purine consists of two rings, pyrimidine and imidazole ring. Now the next slide. Next slide is this one. This one is the differentiation between purine and pyrimidine. So purine is subdivided into adenine and guanine, and pyrimidine is subdivided into thymine, cytosine, and uracil. So this one, adenine and guanine, adenine, guanine, thymine, and cytosine. These four are found in our DNA, right? These four found in our DNA, while in the case of RNA, this thymine is not found. In the case of RNA, this one is not found. In the RNA, adenine, guanine, cytosine, and uracil. I I think you all know about that because you all have read in your intermediate 12th or 10th class also. So adenine, guanine, cytosine, uracil, these are found in our RNA, and adenine, guanine, thymine, cytosine is found in our DNA. Now we have to see the structure of this compound and this compound. So if you want to learn this one, so firstly you should learn this one, particularly chemical name, 6-aminopurine. So if you have learned this structure and the numbering you know very well, then surely you can make at the 6th number you have to attach your amino group. And the guanine, again in the guanine, this one is also a type of purine. So here you have to learn that on the 2nd and 4th number, the 2nd number there will be amino group, and on the 2nd 6th number there will be one ketone group. Similarly here in the case of pyrimidine ring, also in the pyrimidine it is subdivided into 3 parts. So here also you have to learn all the structure in this biochemistry. Don't forget, this one is a biochemistry. Don't think about that this one is organic chemistry, this one is biochemistry, because I know very well most of you are not aware of your syllabus. The next thing, biosynthesis. So we have to learn here what is the biosynthesis of purine and pyrimidine nucleotide. So biosynthesis is a multi-step process which is carried out in the presence of enzyme, and here the substrate are converted into more complex product. This biosynthesis main site is our liver. In the liver, all this one purine nucleotide and pyrimidine nucleotide synthesis held. Next one, this one synthesis of purine nucleotide. It may be synthesized by 2 processes. The first one is de novo synthesis. The second one is Salvage pathway. So this de novo synthesis, the site of action will be a liver and cytosol. The Salvage pathway site of action will be a plastid. Next one, this one is another thing. This one is a salt. This one is, I can say, this one is a trick to learn that in the Salvage pathway, the starting material will be PRPP plus base. Then only you can make a nucleotide. And when the this one, many types of nucleotides will be formed, that will be called as your particular DNA or RNA. Next, in the case of de novo pathway, the starting material will be PRPP, amino acid, ATP, and CO2. Then only you can make your nucleotides. So this one is the small trick to learn this one, de novo pathway and Salvage pathway. Now, this is looking like a very bigger and very tough, but it is not a very tough. Only you have to learn. As I think you all have aware about the glycolysis cycle, Krebs cycle. So similarly here also you have to learn this one pathway. Okay, this pathway you have to learn it, and so how will you learn it? Only firstly you have to learn this one, ribose 5 phosphate. This one is a de novo synthesis of purine. Here you have to learn first compound will be ribose 5 phosphate. Then you have to learn like this only. I'm going to tell you only the trick, and rest only you rest the work depend upon you only. So first thing, ribose 5 phosphate. Then you have to write. Then you have to learn like this, PRPP. Then as first, and you should know the name also, full form of PRPP. That means phosphoribosyl pyrophosphate. So PRPP. The next one here is after PRPP you have to write down ribosyl amine. After ribosyl amine, and if you suppose that if you miss any step, then you can just make here the long this one long arrow, and in the meanwhile arrow you have to make like this. So it will indicate that there are 2 to 3 steps. After 2 or 3 steps you have made finally this one ribosyl 5 phosphate, then formyl glycine ribosyl 5 phosphate. This enzyme, whatever the product is there, that product name you have to write down here, and after that you have to write down synthase enzyme, synthase. If you just learn like this, then surely you can learn it. And after this one, these like this only, finally you will get your inosine monophosphate, IMP. Okay, I'm not going to read this thing. You can read it, and you have to prepare it too for your biochemistry. So you have to learn this one. Enzyme name is so easy. Only somewhere you have to write down synthase, somewhere you have to write down transferase. Okay. The rest thing only you have to learn what the spatial cheese is. Only you have to learn the starting material and your final product. And after the formation of IMP, what will be happen? This one IMP, it will be converted into a adenosine and guanosine. Okay, as you know very well, this one is the biosynthesis of purine. Purine is adenine guanine only. So when this one inosine, it will be converted into a adenosine monophosphate and guanosine monophosphate. Okay, so next thing is yeah, formation of so this one is a similarly after monophosphate formation. It means from the IMP you have made AMP monophosphate. After the formation of AMP you have to make diphosphate, then triphosphate. Okay, firstly mono, फिर di, फिर आ जाएंगे भाई साहब ये triphosphate. Okay, so like this only you have to learn it. This one is another synthesis, purine biosynthesis, pyrimidine biosynthesis. You understand that here I have not to explain. Here only this one is the your work. I have provided this one slide. And now this one is the your work that now you have to prepare it well. Okay, so purine biosynthesis, here you have to learn it. And this one enzyme name also. Finally from the IMP, you can make a ATP and then GDP, adenine and guanine monophosphate, then di and then tri. In this pyrimidine biosynthesis, the first compound will be bicarbonate, then carbonyl biophosphate, then carbonyl aspartate, dihydroortate, orotate, orotidine, then UMP, CTP, CDP. Okay. In this pyrimidine, you have to learn regarding the uridine. You have to synthesize amine and uracil. Okay, it means the final product will be this one, and the starting material you have to learn it. Next one, this one is another slide where the everything is simple. Here you can see the de novo synthesis is carried out from the starting material ribose 5-phosphate, then IMP, then xanthine, then after this one your adenosine, guanosine, etc. And this one is our salvage synthesis also. And the thing you have to focus that from the xanthine it will make a uric acid, we will see. Now here this one is the salvage pathway. In the salvage pathway, again from the adenine, it will be converted into adenosine monophosphate. From the guanine, this one guanosine monophosphate. Okay, the next thing, in the hypoxanthine, you can make this one IMP. Okay, so is it easy now? When you will learn, so everything will be easy. Adenine to adenine, guanine to guanine monophosphate, and hypoxanthine to yes, from hypoxanthine to IMP. Next one is this one is our catabolism. This one is catabolism. So that one was our. Wait for a second. Yeah, so this one is a catabolism of purine. So this one catabolism means, as you know very well, metabolism, as we are discussing here about the metabolism. Metabolism consists of two particular reaction. First one is your anabolism, next one is catabolism. So anabolism means building. Okay, that one is a buildup of particularly your purine and pyrimidine. And this one catabolism means catabolism means breakdown of purine and pyrimidine. So as you know, purine example is AMP and GMP. So AMP, again in the presence of these enzymes, the reaction will be just opposite. From the AMP, it make adenosine, then inosine, then hypoxanthine, and from the GMP, guanosine, then guanine, and the both in the presence of enzyme, it will make a xanthine, and then from the xanthine, in the presence of xanthine oxidase, it will make a uric acid. So now our question is, what is uric acid? So now we have to learn regarding the uric acid. So firstly, before uric acid, we should know that we have read here regarding the biosynthesis of purine, and the pyrimidine. The next one is catabolism. After catabolism, we have to see what are the inhibitors of purine biosynthesis. So first one is folic acid. It is essential for the synthesis of purine nucleotide. So here the inhibitors is, first one is our all the sulfur drugs. It means those drugs which having a sulfur group, like sulfonamide drugs. So that sulfonamide may inhibit the synthesis of folic acid by microorganism. Now what is uric acid? Uric acid is the end product of purine metabolism. Okay, that is when uric acid concentration will be high in our body. That is termed as a hyperuricemia. Okay, that one is termed as a hyperuricemia. It means increased concentration of uric acid. The next one, because of the deposition of this one urate crystal, that's that particular disease is also called as gout disease. Mostly this disease occurs at our toe of this one particular leg. Now this gout is a form of arthritis that occurs as a result of the in the body. So as I told you this, only you have to learn what is uric acid. This one is the end product of purine metabolism. Next one, when this one uric acid concentration will be high, that one will be called as a hyperuricemia. This crystal will be deposited in our particular body. That will be called as gout, and this one gout is a form of arthritis. Now this one hyperuricemia is caused because of the two things, either because of the uric acid synthesis in a high level or the uric acid excretion will be a low level. Based upon this thing, they are divided into subpart, primary, secondary, and secondary. And then because of this one primary hyperuricemia is caused due to the presence of PRPP synthetase enzyme and this enzyme name. Okay, secondary is caused due to the high intake of dietary intake. Suppose that if you are taking more pulse in your diet, then there may be chances of this one gout disease. So start taking everything in a limited amount. Okay, in your protein you have to take in a limited amount, then only you will not be suffer with this one purine metabolism. As you can see, what will be happen if you take more amount of protein, then what will be happen to your body? This, like this. If this one uric acid crystal will be joined here, then you can see. Okay, so these things will be happen if you are taking more amount of protein. So everything you have to take in very limited amount. Now the symptoms, gout start with a sudden onset of pain in one or more joint. Means every time the pain will be occur on the joints only or a toe joint of the foot. Next one, there will be a redness also, swelling also, and the patient doesn't recall injuring the joint before the pain started. Okay. Next thing, mostly this one symptoms occur in the big toe joints, and other common symptoms are in the step of the foot and the knee. And listen carefully, if again this one will be meeting will be closed, then you just be here, because after some time again I will restart and you can join this thing. Okay, so please don't go from here if this meeting is suddenly closed. Then now, how can we control certain foods that are high in proteins, high in purines, can increase the uric acid, high in protein? You should take a limited amount of protein. The next thing, which type of food may cause, especially this one, your gout disease? That one is red meat, fish,